Okay. All right. Well, we're going to first do a summary overview of the kind of metabolism that most organisms, not all, use. Virtually all eukaryotes use these, and of course we're going to leave photosynthesis out of this. We'll treat that separately. But the processes that most organisms are going to use are glycolysis. Literally the splitting of a sugar, specifically the 6-carbon sugar glucose. Fermentation. Not only important for the microorganisms and other organisms that do that, that's billions of dollars a year of commercial activity based on fermentation. And then we have the formation of this very important molecule called acetyl-CoA or coenzyme A. That's very important. Followed by what's called Krebs Cycle, or the citric acid cycle, or the TCA cycle. Very important in terms of generating energy. And then last but not least, I'll call it oxidative metabolism. It's often referred to and is referred to in the text as oxidative phosphorylation. I just think it's a little easier to use the term oxidative metabolism. This process requires oxygen. Now what we're going to see here is in eukaryotes, not in prokaryotes, but in eukaryotes. Most prokaryotes do all this stuff too. The typical garden variety bacteria do all this stuff. But in eukaryotes, the processes I just circled occur in a special organelle or set of organelles. A distinct place where you get all the goodies together in one place, and it's specialized for these types of processes. And that's what we call the mitochondria. And the mitochondria, sometimes called the powerhouses of the cell, produce over 90% of the ATP that the cell uses. Of course, they pump the stuff out into the cytoplasm. But the mitochondria are critically important. Almost all of our ATP comes from these guys, thanks to these processes here. If you happen to be a prokaryote, these processes occur either on membranes in some cases or in the cytoplasm. What we're going to do is look at the mitochondria too before we actually go on to some of these processes. But first, let's overview things. We're going to start out with the process. I'm going to make a long kind of map here. We're going to start out over here. Here's the mitochondria. Let's just change things a little bit. We're going to have the mitochondria over here. All right. Okay, here's the mitochondria. The rest of this stuff's the cytoplasm. Okay, so what's going to happen first? We look at glycolysis. We start out with a sugar. In fact, specifically the sugar glucose, a 6-carbon sugar. And of course, that's produced by photosynthesis. Most of the polysaccharides we consume for food, things like starch and stuff like that, that happens in the innate of glucose. And the process of glycolysis, which literally means sugar splitting. Lysis means to split something. Glycol refers to sugar. What's going to happen is it splits into 2 3-carbon molecules called pyruvic acid. Actually, if we can spell it right. Or pyruvate, same thing. Especially if I can spell it. Okay, interchangeable words. Okay, pyruvic acid is a 3-carbon molecule. Now, in the process of glycolysis, we're going to produce 2 molecules of ATP and 2 coenzymes, NADH. These are these nucleotide coenzymes that can be used to pass electrons from one thing to another. So the vast majority of organisms do use glycolysis as part of their metabolism. Now, we get to a problem here, this pyruvic acid. Pyruvic acid is nasty stuff. I've got a bottle in my lab. It's got a lyric skull and crossbones on, with warnings saying toxic, corrosive, the whole 9 yards. You're making that stuff in your cells. You got to get rid of that stuff. You got to turn it into something you can get rid of easily or something that's less toxic. And one way of doing it is by one of several different processes called fermentation, also occurring in the cytoplasm. What's going to happen is using up an NADH, passing electrons to it in one of the steps. Okay, what's going to happen is we're going to take that pyruvic acid, we're going to tear one carbon off and release this carbon dioxide. And then we're going to have any of a number of different 2-carbon molecules that are both less toxic and easier to get rid of. So, we'll just put 2-carbon. Those 2-carbon molecules can include things like acetic acid, aka vinegar, lactic acid, sour milk, ethanol, booze, and even things like acetone. So all of these things are less nasty than pyruvic acid, and they're easily excreted by the organs. So that's one thing we can do, fermentation to get rid of the pyruvic acid. Okay, now, in eukaryotes, we're going to do the rest of the stuff in the mitochondria. So what I'm going to do is I'm going to move this over here. Just get shifted over a little bit. Okay, now we're going to have the mitochondria. Okay, here's... Okay, here's the mitochondria. Now what we're going to do instead of fermentation, we can do fermentation too, but we can get more energy by further metabolizing that pyruvic acid. In fact, ultimately breaking it completely down to carbon dioxide and water. If we can do that, we'll get far more ATP than we can get from glycolysis alone. That's a good thing. Okay, so what we have to do is transport the pyruvic acid into the mitochondria. Once we're in the mitochondria, we're going to do a fermentation-like process. An enzyme grabs the pyruvic acid, tears a carbon off, releases that as carbon dioxide, and then that 2-carbon molecule that's left, which is ordinarily acetic acid, instead of just releasing it as acetic acid like you do in regular fermentation, this huge enzyme instantly covalently attaches that 2-carbon, what we call acetyl group, acetic acid, to a molecule, something known as coenzyme A. So what we will do is take coenzyme A, oops, and we form a molecule known as acetyl coenzyme A, or acetyl-CoA for short. Now it turns out acetyl-CoA is the fuel for the next part of what we're going to be doing. And that's what's called Krebs Cycle. Yes?
What's the one off the Y, right underneath CO2? What's that word?
Here?
Yeah.
Coenzyme A, or CoA for short. Oh, yes.
What's the word on the other side of the mitochondria?
Oh,
the other side of the mitochondria.
Pyruvic, oh God,
Is
misspelled. Pyruvic acid. Okay, sorry, I mistakenly spelled it. Okay, now we've got our acetyl coenzyme A. Now this is ultimately made from glucose, the sugar. But there's several other ways of making acetyl-CoA, and they also occur in the mitochondria. For instance, we can metabolize fatty acids from lipids, and ultimately it produces more acetyl-CoA. If you happen to use amino acids as energy instead of just using them as building blocks for new proteins, in other words, if you're on a really protein-rich diet like the South Beach diet and things like that, okay, where it's very, very protein-rich, you're going to have to use the amino acids for energy as well, not just building blocks for your proteins, but you have to metabolize them for energy. That process occurs in the mitochondria, and lo and behold, by a variety of different steps, it makes at least the production of acetyl-CoA again. So in other words, anything you eat ultimately ends up as acetyl-CoA. Now, what happens with the acetyl-CoA when you're done with it? Okay, what we're going to do here is we're going to take the acetyl-CoA and we're going to tear the acetyl group, that's 2-carbon acetic acid basically, covalently attached to something. We're going to remove the acetyl group from coenzyme A. We're going to covalently attach it to a 4-carbon molecule and produce a 6-carbon molecule. So let's take the acetyl-CoA and we'll release the coenzyme A fresh. And here's our 2-carbon acetyl group. We start out with a 4-carbon molecule. Add that to it, we come up with a 6-carbon molecule. And then through a series of about 10 steps, we're going to successively break that 6-carbon molecule down. We're going to release, each time we tear a carbon off, we're going to release it as carbon dioxide. So, at some point we go to a 5-carbon molecule. And a CO2 gets released. And then somewhere down the line we go to a 4-carbon molecule. A second CO2 gets released. Now, by the way, this is where we get the carbon dioxide that we exhale. What we're effectively doing is taking that 2-carbon acetyl group and breaking it completely down to carbon dioxide. Well, that's a good idea. It unfortunately doesn't show up in YouTube videos all that well. Okay, now, and we end up after about 10 steps or so with the very same 4-carbon molecule that we started out with. So it's a cycle of enzymatic reactions, and we call this Krebs Cycle. Okay, Kreb Cycle, named after Hans Krebs, the biochemist who figured it out, German biochemist, and also got a trip to Stockholm for that work. Okay, so we got Krebs Cycle. Now, what do we get out of breaking that acetyl group down to carbon dioxide? What we get is energy-rich molecules. At various points in the cycle, let's stick them all in one place, but that's not really true. At various points in the cycle, we're going to generate a GTP, and that can be used to transfer phosphate and make an ATP. So we produce a GTP, but energetically, that can be used to make an ATP, and of course that's more energy. That's one thing we do. We also generate energy-rich coenzymes, nucleotide coenzymes. We're going to produce 3 NADH and one of a similar one called FADH. Now these are where we get the real energy payoff from. Because each NADH, and there's a little debate about this, can be used to effectively produce either 2.5 or 3 ATPs. There's a little debate on it. The FADH is 1.5 or 2 ATPs per shot. The conventional thing is 2 for FADH, 3 for NADH. So there's a bit of a debate about that. Well, we'll probably stick with the 2 and 3, the old traditional thing, but it may not quite be those numbers. But the thing is, that's a heck of a lot of energy you can get from these. The problem is, in order to extract that energy out, now you have to go to our third and final step. And that step is oxidative metabolism or oxidative phosphorylation. Let's put that over here. This process occurs on a series of large enzyme complexes embedded in the inner mitochondrial membrane that's often called the electron transport chain. Because what they're going to do is they're going to take electrons from these coenzymes. They're going to pass them both within each of these enzyme complexes and eventually from one enzyme complex to the next. Each time they do that, now this is oxidation reduction reactions. You pull electrons off of NADH, you have oxidized it. You have to pass those electrons to something. It's one of those protein complexes, enzyme complexes, so that enzyme complex gets reduced. That's exergonic. Energy gets released during that. Now you pass those electrons around to different components of that enzyme complex, so like 20 different proteins in there. Each time you do that, more energy gets released. Pass it from one complex to the next, more energy gets released. Each time you're doing this, you're going to release energy. So what we're going to do here is, we'll just focus on NADH. We tear 2 electrons off. And that generates an NAD+, a positively charged ion of that coenzyme, an H+, and 2 electrons get passed into the electron transport chain. They get passed to the next one. Of course, they get passed around within it. They get passed to the third one. Every time we do this, folks, energy is being released. And that energy will ultimately, by a rather weird mechanism, will be ultimately used to generate ATP. We'll look at the gory details of that later on, but let's just put down here, ATP being produced. Now, the last and final step is this. We have those electrons, we have to pass them to something to get rid of them. What we would call a final or terminal electron acceptor. We have to give those electrons to something. That something we're going to give those electrons to is something that loves electrons. It loves electrons because it's the second most electronegative atom in the universe. And what one is that, folks? Oxygen. The very name oxidation referred to reacting with oxygen because it takes electrons or oxidizes all kinds of things. So what we're going to do is we're going to finally pass those electrons to oxygen. An oxygen atom. And we also have to add 2H plus to this. Now look what we've got here. If you take an oxygen atom, right? You take 2H plus, and then you cancel out the charges with those 2 electrons, you end up with 2 hydrogen atoms covalently attached to that oxygen atom, and that, of course, is water. So we're going to turn oxygen into water by adding H plus and electrons to it. Okay, and here again, when you breathe out, the air you breathe out is loaded up with carbon dioxide, mostly from Krebs Cycle. The air you breathe out has lost about 1/4 to 1/3 of the oxygen compared to what it was when you breathed in. And it's saturated with water vapor. The water vapor is largely coming from this process. Now, think of this, too, in energetic terms. If you take hydrogen gas and oxygen gas, or liquids for that matter, put them together and ignite them, what happens? You get a tremendous release of energy. It's a very exergonic reaction. It's also explosively fast. That's why they use the stuff as rocket fuel. Okay, but energetically, what we're doing is essentially the same thing. We're going to make water from oxygen, but rather than putting hydrogen gas in, we're taking H plus, hydrogen ions, and electrons, adding those separately. But the exergonic nature, the energy release, is the same as burning hydrogen and oxygen. Now, hydrogen and oxygen, gram for gram, pound for pound, whatever, is one of the most exergonic reactions known. That's why they use it for rocket fuel. Ones that are a little more exergonic, like hydrogen and fluorine, are just too corrosive. So, they only made a few experimental, small lab-scale rockets using that. But it's just too nasty and corrosive, not worth the extra energy you get. But essentially, we're taking electrons from these coenzymes, passing them all the way down to highly electronegative oxygen. The energy released from the NADH, passing those electrons from NADH all the way down to oxygen, is tremendous. And that energy can be used to make gobs of ATP. For each glucose, if you just do glycolysis, you only get 2 ATP from it. If you run it through Krebs Cycle and Oxidative Metabolism, you get 2 GTP, that's equal to 2 ATP. 3 of these, 3 times 3, if you use the 3 ATP Figure that's 9. Okay, 1 times 2, that's 11. 12 ATP from here, each time you run Krebs cycle. So, you take a glucose, you get 2 pyruvic acids, 2 acetyl-CoAs. You run Krebs cycle twice, that's 24 ATP. Now compare that to the paltry 2 ATP you get from glycolysis alone. In other words, you gain more than 10 times as much ATP by carrying glucose all the way through these processes as you do with glycolysis. The downside is you have to have oxygen. In the absence of oxygen, you can't do this stuff because you have no place to pass those electrons to. So it just clogs up the whole pathway. So, anaerobic organisms, organisms that live in the absence of oxygen, in fact some of them are poisoned by oxygen, they don't make much ATP. They can't be very metabolically active. They're running on a real tight energy budget. You compare it to people, these are folks below the energetic poverty lines. They can't do too much with that paltry ATP income they're getting. But on the other hand, you use oxygen in metabolism, you have these things, now you've got energy to burn. You can make more complex, more metabolically active cells. Now some of our cells, the neurons in our in our brain, burn through so much ATP they can hardly keep up. They're just producing barely enough to keep them going. That's why you lose consciousness quickly. Sorry. Sorry about that. All right. So that's a skeletal overview of the whole shebang. Well, probably leaves a lot of confusion, right? Yeah. Okay, we're going to look at these things bit by bit, but that skeletal overview kind of helps out. We're just getting the big picture. Now we're going to look at this stuff bit by bit in just a moment. Don't worry, we will come back to all of these in more detail. Okay, this is just a quick and dirty. All right, let me do part 2 here.